Excellent. Wow, it looks like a lot of people are waiting. This is great. Um, so in the interest of time, I will go ahead and start to give some introductions. Thank you everyone for coming to this next session of our BEAD Core Winter Seminar Series. Today, Dr. Sam Vanderhoek and myself will be talking about dysglycemia and pediatric surgical outcomes. And this is both really interesting from a science perspective, what was actually done and what the findings were. And we're also kind of using this as an example of how the BEAD Core can support researchers, because it's a good example of what actually happens when someone is trying to use clinical data to answer a question and the steps that we have to go through to try to answer using data that was never kind of meant for research. So just as a quick housekeeping, if you have questions, feel free to put it in the chat or call out if you would like to add something or ask something, or there'll be plenty of time for questions afterwards. It's a relatively short presentation. I also just want to give a quick plug for the BEAD Core. This is, in case you're not familiar with us, the Biostatistics, Epidemiology, and Data Management Core. And we provide a very wide range of research support services for faculty throughout the institution. We are supported by Department of Medicine, Pediatrics, ACCM, and Gyne-OB, as well as a pilot program with PM&R and a program where we help with patient-reported outcomes. And so faculty in the departments that I named and faculty working on patient-reported outcomes get some supported hours to work with us every year. Please just email beadcore@jhmi.edu if you're interested, and you can submit a brief intake form and we can find someone to talk to you about your project. So without any further introduction, Sam Vanderhoek is an assistant professor in the Department of Anesthesiology and Critical Care Medicine working in pediatrics, and I'll let him go ahead and start with our presentation. Welcome, everyone.
Awesome. Thank you, Laura. And thanks so much for having me. So today, like Laura said, we're going to be talking about a project that we both worked on together, a real collaboration between the BEAD Core and the Division of Pediatric Anesthesia within ACCM. I'll kind of jump to the chase here, which is the background and the question. So I'm a pediatric anesthesiologist by training, and my research involves perioperative glycemic control. In other words, how blood sugar and derangements in blood sugar affect outcomes in pediatric surgery. There are lots of studies about this question in the adult literature, specifically, do abnormally high or abnormally low blood sugars affect outcomes after adults have anesthesia and surgery. And this tends to be studied very heavily in the cardiac surgical literature because there's so much available blood sugar data that can be analyzed. So as you can imagine, in adults who are undergoing heart surgery, there are lots of invasive vascular access for which they can draw blood sugars and analyze. In adults, either adults who have diabetes or adults who don't have diabetes, who have undergone cardiac surgery, elevated blood sugar tends to be associated with increased morbidity and mortality. When we look at the same question in adults who are having non-cardiac surgery, so these can be any sort of surgeries like neurosurgery or abdominal surgery or orthopedic surgery, that elevation in blood sugar is still actually associated with longer hospital and ICU stays and things that you don't really want to happen, like postoperative pneumonia, sepsis, kidney injury, and myocardial infarction. There was a really nice study that showed that the mortality goes up directly in proportion to how high the blood sugar goes up in non-diabetic adults who are undergoing non-cardiac surgery. But what about in kids? What about in pediatric surgical subspecialties? And really, the studies that look at this question in kids are limited to children undergoing cardiac surgery and neurosurgery. And the reason is because the same reason why we have a lot of data in adults, children who are undergoing either brain or heart surgery tend to have a lot of vascular access, and we can draw blood sugars and analyze them. So what do we see in these two surgical subspecialties in kids? So in children who are having heart surgery, what they actually showed was that elevations in blood sugar either during cardiac bypass or after the surgery is over in the cardiac ICU, these tend not to be huge risk factors for postoperative morbidity and mortality. And there have been a couple of subsequent studies that have confirmed this finding. What about in neurosurgery? Children who are having neurosurgery, we see the opposite, actually. So we see that they have longer hospital and ICU stays and higher risk for postoperative complications when blood sugar is elevated. And this is something that's actually been shown in the ICU literature as well. So the question we really wanted to ask was, well, what about everyone else? What about all these other kids who are not necessarily having heart or neurosurgery, but just having some other surgery, orthopedic surgery, abdominal surgery, ear, nose, and throat surgery? Do they have a higher risk for a bad outcome if they have dysglycemia, which is a fancy word for either high or low blood sugar during the perioperative period? So it seems kind of like a very basic question, but the devil is in the details. So how do we get the data? So the American College of Surgeons runs two large databases. One is the National Surgical Quality Improvement Program database for adults. And then it has kind of like what I call the stepchild, which is the pediatric version of it, which is still codes for the same things, but I think it's used much less rigorously. So what happens is clinical abstractors at multiple institutions collect standard demographic data, preoperative risk factors, baseline medical conditions, intraoperative events, and lab studies, postoperative outcomes for both inpatient and outpatient surgeries in the pediatric community. And each operation is associated with its CPT code, the current procedural terminology code, and it's sorted by surgical specialty. The database collects 18 individual postoperative adverse events as dichotomous variables, and these events are followed for the first 30 postoperative days. What's nice about this data set is that there's no missing data. The database excludes cases coming from institutions with a 30-day follow-up rate of under 80%. What is one thing it doesn't include? It doesn't include blood glucose data, which is, of course, exactly what we wanted. So Hopkins, both the downtown campus in Baltimore, as well as All Children's in Florida, is a contributor to the NSQIP database. So what we did is we took the Hopkins contributions to the NSQIP database at both sites and essentially re-identified the data and matched those surgeries with all of our blood sugar data in-house in our own electronic medical record. So for Hopkins, that's in Baltimore, that's Epic. And for All Children's, it's currently Epic, but up until about a year and a half ago, it was Cerner. So we essentially matched the encounters from the NSQIP database to the perioperative records in the Hopkins system. And by doing so, we were able to extract all the blood sugar data within a 9-day perioperative period. And those 9 days consisted of the 3 days preceding surgery, the day of surgery, and the 5 days following the surgery. And we thought that 9-day period was reflective of the larger perioperative window. So again, this seemed like an easy question, but it actually involved 3 very large databases that we had to merge together. So has the NSQIP database ever been studied here at Hopkins? So the answer is yes. And in fact, the pediatric NSQIP database has been studied pretty extensively by our pediatric surgical group, as well as by Emily Boss, who's a pediatric otolaryngologist and ear, nose, and throat surgeon. And she actually used the database to investigate specifically postoperative adverse events and morbidity after ENT procedures. And what she did is, in addition to looking at those individual 30-day adverse events, she developed 3 composite scores, which she defined as composite morbidity, composite serious adverse events, and composite hospital-acquired infections. And really, these procedures and groups that she put together was kind of a way to really get a bird's eye view of how whatever variable you're looking at affects these 3 areas. So composite morbidity really was if you had any one of the adverse events. These are things like pneumonia, being intubated for a long time, having a cardiac arrest, et cetera. Composite serious adverse events is just a more high acuity postoperative problem. So these would be things like death within 30 days, reoperation within 30 days, which is really not a good outcome, or being in the hospital at the 30-day mark, so a long admission. And then the hospital composite acquired infections is kind of what it sounds like. So these are if you had any sort of surgical site infection, urinary tract infection, or pneumonia within that 30-day period. So we really used Emily's model because it was a very useful model to analyze postoperative outcomes and how they're impacted by dysglycemia. I'm going to turn it over to Laura now, who will talk a little bit more about how we managed this very large data set and how we subsequently analyzed the data with various statistical methods.
Thank you. So as Sam said, it was a very complicated set of data sets that we then needed to merge together. And given that a patient could have more than one surgery theoretically in the data, it wasn't possible to just merge, for example, on MRN. We had to also look at the data of the surgery to make sure we were matching the correct surgery and the correct person. And this is always made more complicated by little things like, strange metadata attached to certain data locations. And there were some data formatting acrobatics that had to happen, but that is what we're here for. So we did manage to merge these all together into one usable data set, though I will say that was a really complicated process for sure. And to sort of get a bird's eye picture of what in general the idea of transforming clinical data into analytical data looks like, I am happy to share if anyone wants to reach out to BEAD Core at jhmi.edu. Bahar and Modanlu's wonderful presentation. It was, I think, our most recent winter seminar presentation about big picture, what does it look like to use EPIC data or PMAP data for research? She did a really good job of explaining all of the complexities and conversations that need to go into this process. But big picture, this kind of collaboration between clinicians and data specialists is really essential to making it work. And, especially when it comes to things like creating these composite outcomes, it's a great case scenario in any kind of research when this sort of an outcome is already published, it's in the literature, it's accepted as a great way to look at these outcomes and use clinical data in a way that's really efficient and powerful. But as Sam said, the devils are kind of in the details sometimes to create those from our own data. Next slide, Sam. Okay, so this is, I know, a mess, and I don't expect anyone to be able to read this and process this information. I was just trying to kind of give an example of what our working document looked like for this study as we were trying to figure out the real nuts and bolts of how we were identifying the data elements that needed to be used in this study. So, for example, what Sam started with from the original data is column 3 and 4, I think, the variable name and the variable label. So just loading things into Stata, that is the variable name that I was looking at. So looking at this first one of post-op search superficial incision, I need to figure out what does that mean? And I've got this label that we can add from the original code book, but then it needs to be formatted in a way that it makes sense for the outcome that we're interested in, and also to create those composite measures that we're interested in. And so after going through all of this data, with some original notes from Sam on the left-hand side of what the characteristics are that we're actually interested in, we went through just a series of columns of questions, like, for example, looking at I'm saying, is this right? Is this right? But do you want yes or no for these? This is a continuous number. If you want this to be yes or no, what should my cutoff be? And things like that. And that's just really typical of how these kinds of collaborations go. So this is not as much of a train wreck as it looks like. It's actually what was helpful to get things moving along. So next slide, Sam. Thank you. So as I said, the formatting was definitely the hardest part. In the end, for the analysis, because we were dealing with 2 institutions and we were dealing with patients who could be in the data more than once, we used a mixed multi-level logistic regression model. And we're looking again at the relationship between these abnormal blood glucose measurements and these adverse postoperative outcomes. And the multi-level model let us use sort of nested levels for both the patient and for the institution, because we would assume that sort of within a patient, you might have some of the same characteristics and outcomes and the same goes for within institutions. And so we were able to handle that sort of lack of variability that way. We also later ran stratified models for the most common surgical specialties. And this is something I recommend doing if you are dealing with sort of disparate groups, which these different kinds of surgeries might be, to make sure one of them isn't completely driving your results, or to make sure that your null results aren't the result of a positive relationship for one type of surgery and a negative relationship for another type of surgery, for example. So we just wanted to dig in and make sure we really understood what was happening. And in the end, when general pediatric surgery seemed like it was a driver of the results, we further stratified by the procedure group. And that was another back to the drawing board. Well, what are the CPT codes that identify that procedure group? Just make sure that we're having a complete list and we're all on the same page about how to identify those. And so those were sort of that's the bird's eye overview of what our analysis looked like for this project. So I think we go back to you now.
Yep. So what did we find? So what we found, which was not entirely surprising, was that children with dysglycemia, again, either a high or a low blood sugar, are more likely to have a bunch of adverse events, surgical site infections, cardiac arrest, sepsis. They're more likely to undergo reoperation, and they're more likely to remain hospitalized at 30 days. And this was after controlling for patient demographics, for case demographics, including surgical length, hospital site, et cetera. What we found was that dysglycemia was associated with more composite serious adverse events, more hospital-acquired infections, and a higher composite morbidity compared to patients who had a normal blood sugar. Like Laura was talking about, we wanted to make sure that one surgical subspecialty was not driving the results that we were seeing. And we looked at essentially all children who were undergoing non-cardiac surgery, because cardiac surgery has been studied pretty extensively in the literature and how dysglycemia impacts postoperative outcomes. So we were looking at multiple different surgical subspecialties, including general pediatric surgery, neurosurgery, ear, nose, and throat, and then a lot of kind of smaller surgical subspecialties like plastic surgery, interventional radiology, our PICC team for putting in invasive PICC lines, et cetera, et cetera. And when we kind of stratified, we found that the highest association with perioperative dysglycemia and adverse outcomes was in general pediatric surgery. And then the next logical step is, well, what specific surgical procedure within the realm of many surgical procedures that general pediatric surgeons do, is there a particular procedure that we tend to see the highest risk? So we essentially took all of the NSQIP data, sorted it by CPT code, and then really tried to make fairly specific groupings of the types of surgical procedures, because while yes, a congenital diaphragmatic hernia is a congenital diaphragmatic hernia, there's 30 or 40 ways to code it in the CPT system. So we had to kind of like put them all together in bins and then analyze it that way. And at the end of the day, children who are undergoing an anerectomy or colectomy seem to have the highest association with a serious adverse event, which is not surprising because typically if you're having an exploratory laparotomy, your belly's being operated on and your bowels are being manipulated, this is a surgery that involves a lot of fluid shifts, probably a lot of electrolyte abnormalities, and any sort of associated dysglycemia would probably not be super well tolerated as just another variable within the perioperative recovery process. So we will turn it over to questions, and Laura and I are happy to answer either nitty-gritty about this particular project or kind of how this partnership / collaboration worked on the ground.
Feel free to just jump in with comments or questions.
Hi, Laura. This is Priya. Great talk, and I have done some work on the script too. So it's good to see that you guys like merged Niscript with Epic and stuff. So did you have to go back and create, like, did you create a new variable from the Epic, or how did you do it? Like, did you just get the MRNs? Like, how did it work?
Yeah, so what we did is we re-identified the Niscript data, got the institutional MRN, and then using help in the Department of Surgery, we're able to go into both Epic and Cerner, because Cerner was the medical record at All Children's until last year, and then pull all blood sugar data in this 9-day perioperative window. That kind of window we defined for ourselves. But basically all blood sugar data was pulled with the date and the time. And then we were able to map the blood sugar data, whether it was at 3 days pre-op, 1 day of the surgery itself, or the 5 days post-operatively. And kind of put everything into one record for each unique surgical procedure, with the understanding that, like Laura said, there were some patients who had multiple surgical procedures as part of the data set.
Right.
Does that make sense?
Yes, it does. Also, Dr. Vanderhoek, like, how long did it, like, how was the procedure to go back, like getting data from Niscript and going back to Epic? Like, how complicated or easy was that?
It wasn't that complicated. We essentially looked at a 5-year period, and we started the period in 2016 because that was when the bulk of the Hopkins Baltimore campus went on to Epic. I think we could have gone earlier, but that would have involved using another electronic medical record to pull blood sugar data from, and that seemed just a little onerous because 2 was already hard. But once we actually got the data with the timestamps of the blood sugar, it was fairly easy to then map it out. And as you can imagine, there were some patients who had only 1 or 2 values, and there were some patients, usually the sicker ones in the ICUs, who had upwards of 50 or 60 blood sugar values. I think the median blood sugar, the median number of blood sugars was, I think, like 1.5. So definitely most patients just had 1 or 2 blood sugars. But we were able to then gather that data and put it all together. But definitely having 2 different electronic medical records made it a little bit more complicated. The other thing that I didn't realize until I was in the weeds here was that there are many ways that blood sugar data funnels into Epic and Cerner. It's not just a blood glucose. It could be a blood glucose from a venous blood gas. It can be a blood glucose from an arterial blood gas. It can be a blood glucose from a comprehensive metabolic panel. It can be a glucometer, which is, a finger stick. And when I originally used, this colleague in the Department of Surgery to help me get the blood sugar data, he said, that's not a problem. Just pick of these 98 different ways that Epic classifies blood sugar, pick the ones you want me to pull. And, there's some glucose data in that set, which were urine glucose, which was not relevant, or glucose from the CSF in a spinal tap, which was not relevant. But I don't know, it's like, you don't really think about that stuff until you're actually looking at this list and you're like, oh, this is actually more complicated than I thought. And then there's some, sugar, glucose data, glucose variables in Epic, which have no associated collection source. So if someone enters an order for a glucose and doesn't specify the source, it just funnels into Epic as glucose. And it's up to the provider to remember, oh, that was actually from the blood, or that was from the urine. But those values we kind of had to throw out because we didn't have that context.
Right. That's really intensive though. Dr. Vanderhoek, another question. So was it all had like all these values had one code, or does it had like different type CPT and some other kind of codes too?
Do you mean the surgeries?
No. Like so there is this OMOP data model that has, as you're saying that, you used blood sugar level from different sources. So it was that the description of the test, as you say in the
Correct.
Epic, or was it like codes?
No, it was the description of the test in Epic.
Okay.
Yeah.
All right. Thank you. That's informative.
Sure. And I think in the chat, Johannes is just asking if this project is on preoperative or perioperative dysglycemia. So the answer is it's perioperative. It's 3 days pre-op, the day of surgery, and 5 days post-operative. So it encompasses a 9-day perioperative window. And then Amanda is asking if we know of any discrepancies in how different sources of glucose are read. That's a really good question. It's actually something that I've studied in a different project. But we assume that the data that goes into Epic is fairly well validated by clinical engineering to be a relatively high fidelity, marker of the glycemic status of the individual. So there can be discrepancies, like if you get a finger stick in someone who's extremely clamped down and the source of, interstitial space that you're sampling might not be reflective of, the patient's actual blood glucose. But I think those are pretty few and far between. So we kind of made the assumption that, the blood sugar value, whether it was obtained from a venous stick or a glucometer, they're fairly similar, but that is a limitation of the study because not every blood sugar is going to be the same as the corresponding finger stick from a glucometer. Charbel, you have your hand up.
Doctor, first of all, amazing project, and came right in time because I'm the new postdoc with the General Pediatric Surgery Department, and I work with NSQIP, and we have a new project about hypo or hyperglycemia in neck. So I have just a few questions for you. You said that you took the glucose level for 9 days. How did you compare them? Did you compare them 1 day at a time, or just like the overall value of them?
So we looked at the 9-day period, and essentially anyone who had an abnormal blood glucose within that 9-day period, be it high or low, they were put into a dysglycemic bin.
Okay.
And anyone who had essentially a normal blood sugar was put in a euglycemic bin. and anyone who had no blood sugar was not included in the study. So you can imagine we started out with, I believe, Laura, we started out with about 12,000 records from NSQIP,
I think so.
roughly half and half between all children's and Baltimore campus. And 7,000 of those records had no associated blood sugar, which makes total sense, right? Why would a healthy child undergoing a laparoscopic appendectomy ever have a blood sugar checked, right? It's not super common. So 7,000 of those 12,000 cases were excluded, leaving us with a little more than, I think, like 53 or 5,400 cases that were included in the project. But no, we did not look at when that abnormal blood sugar, if it was obtained, when it was obtained in that 9-day perioperative window.
So the variable of the outcome was categorical, not continuous. So it's either dysglycemic or euglycemic. You did not look like at the specific level of blood sugar, right?
Exactly.
Okay.
We did kind of some preliminary analyses up front, looking at hypo versus hyperglycemia, but we ended up kind of not going that route.
Okay.
The
And
other last thing I was going to say is adding an additional layer of complexity to this project, which Laura and I kind of had to deal with in the middle, was neonates. So in other words, babies who are in the first few days of life. A normal blood sugar in that patient is actually very different than a normal blood sugar in a baby who's more than a few days old. Specifically, we typically define low blood sugar as under 70 or 60, but in a baby who's under 96 hours old, a low blood sugar is under 40. So we would have actually categorized these babies as being hypoglycemic if we didn't adjust their limits of normal down.
And for each day, for example, when a patient had multiple blood glucose drawn from the same day, did you just take every single value of them, or did you just choose the median, the average of blood glucose for that day?
We considered every value.
Every.
Yeah.
Thank you very much.
Of course. Any other questions?
I'll ask a quick, more basic question, which I think we get from a lot of our faculty, which is, do you need to have any baseline experience as a clinical researcher, data science, et cetera, to interact with the BEAD Core? What if you just have a study idea and that's all you got and you've never kind of fully explored? I guess it's a question for our BEAD Core colleagues. How can you just say, hey, this is what I've got and where do I go next?
Sure. Well, first I'll say we love getting started on the ground floor of studies. When studies are being imagined, you know, we would much prefer to come in at that point rather than come in once you've collected data and maybe it could have been done in a way that made it a lot easier to work with, or your study design wasn't entirely thought through, something like that. So we do, for point one, prefer to get in on the ground floor. Two, our support services are also extended to residents and fellows, provided that they're working with a mentor in a sponsored department. And we do sort of, because we're not necessarily content experts in whatever particular area of clinical science that we're working in, we do request that the mentor attends the initial consult and remains involved. So we will definitely help provide that level of data support, stats support, study design support, but we would like to do it in guidance when it's someone who's relatively new to research. We'd like to do it in guidance with a mentor who can help make sure that things are solid on the content side of the science.
Great, thank you. Thanks, Sam and Laura, for an awesome example of how that all comes together. Thank you.
Absolutely. Any other questions?
I got a request via the chat to share. I guess my question was a little loaded because I've interacted with you all and you're fantastic. And Dr. Vanderhoek's experience reflects that really beautifully. And I've worked with Dr. Vaidya and numerous other colleagues, and I came in exactly that same way. I had no clue what I was doing, and I just had an idea, and I wanted to build a REDCap database, and I needed statistical help, and these were all things that I didn't have a starting point for. So I just wanted to put in a plug for just incredible collaboration opportunities here, and that can lead to publication and all sorts of other things. So grant writing, et cetera, which were nicely illustrated here.
Thank you. Thank you so much for the support. And anyone who wants help, again, just email beadcore@jhmi.edu. And any other questions? All right. Thank you all so much for coming. Feel free to reach out to BEAD Core with any questions. We will also have a recording of this and the slides. So feel free to reach out with any questions or requests like that. Thank you so much, Dr. Vanderhoek, for being a part of this winter seminar series. We really appreciate your time, and thank you all so much for coming.
Of course, thank you.
Thank you. Have a good day.